Department of Zoology as well. And the main question of our research group really is how did the diversity we see nowadays evolve? And of course, we can't study all possible animals on the whole planet, so we often focus on one model system, right? And ours is cichlid fishes. And so what we try to do is really look at the mechanisms that underlie evolutionary change. And what I mean by this is how do these organisms become different from one another? And the approach we take is we look at their DNA and we try to find which bits are different. And then we compare how do these different forms develop from egg to adult form to try and see what is the impact of the differences in their DNA. How this affects adult trait variation, and then how does it affect how organisms interact with the environment and with one another. And we do that by studying cichlid fishes. And cichlids, I think the three major radiations come from East African lakes. And this is a picture from Lake Malawi, and they are really, really vast. They don't feel like actual lakes. When you're sitting next to them, you feel like you're by the sea. And then under it, here it's a video from planet Earth. And under it, it's what's even more dramatic, I feel like, because just in this one lake, there's 850 species that evolved within less than 1 million years. And they differ in color, head shape, lots of different traits, you name it. They're all very different. But the most fascinating thing is that genetically, they are very, very similar. So there's almost as much genetic diversity between human populations as there is between these cichlid species, right? And so they are kind of a natural mutagenic screen, let's put it that way, for us to study how do species become different from one another. And so here is just one slide to show you how different they can be. So you have differences in head shape there on the left, body shape variation, fin shape variation. Also, the craniofacial skeleton is very different. And not just morphology. In terms of physiology and behavior, they're also extremely different. And so you have a huge community of evolutionary biologists looking at these fishes and really trying to understand how does such rapid diversification occurs. And so here is one of our cichlids, Astototilapia calyptra. In fact, this specimen is very important to the lab. It was the first fish male we actually had hosted in our fish facilities. And the lab focuses on pigmentation, and in particularly these egg spots, which are the circular markings on the anal fins of about 1,500 species of cichlid males. And they are a sexually selected trait, so females tend to prefer males that have more egg spots, or sometimes it's sexually selected indirectly due to male-male competition. So the male that has the better body condition has more egg spots and will hold the best territory. And they're actually quite important on the breeding behavior of these species. So you see the big male courting with the big female, and they go round and round. The female is laying the eggs, you see them here, and she immediately takes them up in her mouth, but she's not eating them. These fishes, they're mouth brooders, and so they will hold the eggs in their mouths until they can swim independently. And after collecting the eggs, the female will nibble on the egg spots of the male, so they're also called egg dummies. And this is when fertilization occurs, so the sperm gets released and fertilization is internal, away from all the predators, because you've seen the density in these lakes, right? Then the female, it's a huge effort. She won't feed for the next 3 weeks while holding the eggs. And here I have another BBC Planet Earth video just to show you this mouth brooding behavior in these fishes, which is quite extraordinary. So now they're swimming independently, but they will still hide in the mother's mouth if they need to. So you see them flocking around, whoop. Exactly, it's fascinating, isn't it? Yeah. And so why do we focus on these egg spots? So they are a very important trait, sexually selected trait, and also important for the behavior of these fishes. But they're also really diverse. And so we can use it as a model system to study how this trait becomes different between species. And here on these first three columns, we have diversity between species. And here on this one row, you have diversity within a species. So you can study different levels of diversification. And using the cichlid diversity, you can also study different evolutionary events. So here you have the origin of egg spots. So you go from a fish that doesn't have this trait to a fish that suddenly has it. So we're trying to see how these novel traits appear, and then how they diversify both within and between species. And I'm not going to go into much detail into the results, just give you like a flavor of what we do. So here we were interested in trying to understand how these spots emerged. And so what we've done was to compare the genomes of species that have egg spots with species that don't have egg spots. So in these little schemes here, you can see that the species, in fact, doesn't have much pigmentation on their anal fin. Then you have some species that have sort of this blotch, and species that have the actual egg spots. And what we found, for example, is that in this gene that actually acts on a pigment cell, the species that have egg spots have this bit of DNA, which is a transposable element, for people who are interested in knowing what it is. But this DNA bit doesn't exist on the others. And so like this, we could associate the presence of DNA variation to a trait, right? But then we also look, as I said, to diversification within a species. And here we are looking at a smaller lake. So it's very small, so you can actually see the other side of the shore is not as vast. And it's a little crater lake called Lake Masoko or Kisiba. And here, you can see also, how it's located just north of Lake Malawi. And here we don't have as many species, but we have two populations of the same species that are just starting to diverge from one another. And so basically, it's as if we caught the speciation process just happening, right? Which it's very, very rare. And so you have this shallow population that it's yellow and lives from 0 to 5 meters. And then you have this very deep population, 20 meters, which is blue. And here is just a representation of the light environment. So on the deep, on the right, and on the shallow on the left. And I hope you can see that, so the axes are very different. So here it goes up to 1,000, and here it goes up to 100, and this is light intensity. So you see that on the shallows, they have a lot more light. And then on the deeps, what happens is that there is a narrowing of the light spectrum. And so basically, there's low light, and the light that gets down there is green and blue shifted. And so what this system allows us to do is actually to see how these species adapt to, so it allows us to see the why they are adapting, because the two environments are drastically different. So they have to adapt to this dark, cold, low oxygenated, and very different in terms of prey or diet, for example. And because it's happened very recently, so only in the past 1,000 years, what we can really see then is exactly how they're going about it, right? So how are they becoming blue? How are the anal fins changing in color? And then related as well to their visual system and to their dietary preferences. So this really helps us address the why and the how questions. And so we're also looking into the genetics of this system. And so I hope it was all very brief. Sorry, I didn't go into much detail in terms of the actual results. But I hope I could give you sort of a flavor of what we do with these fishes and how to go about the mechanisms underlying evolutionary change. And yes, I just wanted to show our research group. and also a lot of the collaborators, both here and in Tanzania, where we do a lot of our work. So yeah, thank you.